Hi, I'm Terry Virts, and this is Down to Earth with Terry Virts, where we talk about what matters down here on planet Earth. And today I have an awesome guest, Dr. Jonathan Kurtis. He is a medical doctor, he's a PhD, he's a Stanley M. Aronson Professor and Chair of the Department of Pathology and Laboratory Medicine at Brown University, which means he's an underachiever and he runs the department that studies the mechanisms of disease. And he's been performing research on a malaria vaccine for over 20 years in Africa. And Jake, it is so awesome to have you. Thanks for joining me.
Hey, my pleasure, Terry. You're right, man. I'm a total slacker. It's been my MO. My mom now feels vindicated. Someone's called me out on it.
You're the black sheep at the family Thanksgiving dinner.
Exactly. It's terrible. What's going on?
Well, some of the guests I have on here, I feel like, what have I done with my life? But so thanks for having me on. We met about a year ago through some medical. I was trying to help out with some different medical things, and I was just fascinated and amazed at the work you're doing and how important it is. Obviously, shortly after we started to get in touch, there was some disease that happened last year. I can't remember what it's called, but that kind of took the focus.
Disease. Let me just clean myself up with a spray bottle.
There's the college professor prep.
That's awful. Awful.
So are you teaching college classes now, or do you do mainly research?
Yeah, so my life is kind of split. I spent the first 15 years of my, when I was finally getting paid by Brown instead of going to Brown. I spent the last 15 or so years spending about, a third of my time to half of my time seeing patients, doing clinical medicine, and the rest of the time doing research and, teaching a few lectures here and there, but teaching wasn't a huge component. In my current job, I've got an administrative burden, which is, I sometimes feel it's Sisyphean, and I'm never going to get that rock up the hill. And a little bit of clinical work, but then mostly research. So I think, the research has always been the main thrust of what I've been trying to do.
Okay. So first of all, Brown, like growing up, I grew up in Maryland, and I knew that Brown was somewhere up to the north, and I knew it was a big, Ivy League school. Where is Brown?
Yeah, so we're in Providence, Rhode Island, the cultural mecca of the Northeast. Forget about New York. Forget about Boston. Actually, it's a really beautiful town. It's a town of about 100,000 people. The state has a convenient denominator of 1 million. So if you're an epidemiologist, you can do the math in your head when you're dividing. Lots of coastline. It's actually really cool. And I don't know if you saw I think it was in the Democratic National Convention where the local yokel from Rhode Island called it the calamari state. Do you remember that?
It was a riot.
No, it was like every Rhode Islander was just going, oh.
That's pretty funny. So you have an MD and PhD.
Yeah. What's up with that?
Yeah. What kind? In what flavors?
Yeah. So the MD is sort of just the standard garden variety MD. I did residency in residency in clinical pathology.
Okay.
And then fellowship training in transfusion medicine and coagulation. So blood banking and coagulation is sort of my gig. You pretty much don't want me as your doc. I've actually gotten in fist fights with priests. They're trying to get in the room to go for the last rites. I'm like, they're not dead yet. Remember that line from Princess Bride? He's not dead yet. He's nearly dead. I'm that guy. I'm Billy Crystal.
And how about the PhD?
Yeah. So the PhD was in molecular parasitology. It was kind of a funny story.
That sounds like a funny topic.
No, exactly. How the hell that? No, it's actually true. So I was a total, not that I've gotten any better, but I was a total academic loser in college. I didn't find my mojo. I had no sort of direction. Brown is a great school insofar as enormous amounts of intellectual freedom. It's designed for individuals who are truly academically self-actualized. Which I guarantee you was not me at age 19 or 20. So I was wasting my time. And I actually, I have this sort of depressogenic theory, if you're not achieving the thing of your age strata, whatever that may be, it sort of throws you into a funk. And so my friends, I had some great friends, and they were all just ripping life off the bone in big chunks and eating it. And really, you could see them moving. And I felt like I was in a miasma. So in that miasma, I looked up to a friend of mine. Her name was Monica Brady. And you may actually have heard her name. She went on to become a correspondent for Voice of America and then NPR. She helped break the Catholic priest scandal in Boston. She was part of that whole team that brought those nasty guys down. Work that still needs to continue, I might add. So anyway, Monica spent a summer abroad in Kenya, her junior year. And she came back with unbelievable stories. And so I thought, hell, I'll do that.
Right.
Which, that was the depth of my investigation, pretty lame. Anyway, I got lucky enough to have two things happen to me in Kenya. One, I fell in love with research. I was doing a research project on the coast of Kenya. It was actually population ecology of a sea urchin. So it was actually coral reef ecology, but it was real research. It was not demonstrations in classrooms. It was trying to discover new stuff. And I loved that. Yeah. And then I also got cerebral malaria, which normally you think is a bad thing, right?
Right.
But it was a huge eye opener. I realized I loved research, and I also came to realize that malaria was the great thumb pressing down on a population. No society could possibly flourish if a quarter of its children were being culled before the age of 5. I mean, it really brought it home to me. Anyway,
that's in Kenya.
I go back. Yeah, in Kenya. I was on the coast of Kenya.
Right.
Yeah. So I go back to Brown, and I suddenly knew, I thought, what I wanted to do. And I was in the MD program. Brown had an 8-year MD continuum, 4 years of undergrad, 4 years of med school. So I started med school, and I freaking hated it. I hated it. I mean,
why?
Super. Well, Brown's this really creative liberal college. Right. Everyone there is fabulous at some weird ass thing, like, playing the oboe in their sleep. They all have these ridiculous talents.
Right.
And they're all interesting, and they're all different and diverse. And then you get to med school, which is the giant frappuccino of the educational continuum. You take all these interesting people and you put them in the blender, and you go, nah, and you turn them all into the same mush, because they take the exact same class. And it's an unfortunate reality. Medicine has hundreds of years of experience treating disease. A lot of what we do actually works. And so if you have pneumonia, you want to be treated exactly the same way as everyone else with pneumonia, because 99% of the time you're going to survive it. You don't want somebody going, I wonder what I could do here.
I don't want the creative doctor. Yeah. I want the
until you've exhausted, until you've exhausted what we can do. So anyway, you turn all these interesting people into zombies. I hated it because it was lots of memorization of other people's science. It wasn't science. So I went in to quit. I had a mentor figure. His name is Dick Old, who I was actually just talking to a couple of days ago. I went in to see him. He was the guy that treated me for malaria when I got back to the States. So he was sort of almost like a father figure, really. I went in to tell him I was going to quit med school. He was a physician scientist. And I'd never heard the man curse before. But he said, "Are you out of your mind?"
Right.
I bleeped that out for you.
That was pretty good.
Thanks. He said, "Don't quit, double down." And I'm like, what? What are you talking about, double down? He said, if you're missing the science in medicine, do an MD PhD, do your PhD in my lab. He worked on a different tropical pathogen. He worked on something called schistosomiasis, which is a nasty worm infection in the tropics that rots out your liver. And so I thought for about 30 seconds, and boom, did the PhD with him. So that really was it was a turning point. Kind of it sort of pinpoints how, your life has, hours and hours and hours of boredom, and then moments of sheer panic. This was one of those inflection points where it could have gone either way. So I'm eternally grateful that he cursed at me.
That's an amazing story. So is that extra time? Did that require extra years of work?
It's an undefined amount of time, which is what gives people pause. For me, it took another 4 years, but they were the greatest 4 years of my life. I met my wife. Her job was awesome. So she was an undergrad in the lab. So these
Okay.
schistosomes, these worms, there's a male and a female.
Okay.
And the male's got this thing, it's called a gynecophoric canal, basically love handles. But on a worm, they're sexy. And so the
Worms dig
female
that.
worm lies, exactly, totally. The female worm lies in the groove of the male. And
Right.
check this out, man, 24/7, they're going at it. Like, doesn't that make you feel inadequate? They are literally 24/7 copulation. You know, not even a cigarette break. I mean, they are full tilt.
Wow.
And
So there's
yeah,
no
so her
worm
job,
Viagra required.
no, not needed, not needed at all. So her job, Jen's job as an undergraduate, is she would take these worm pairs in culture and she would rip them apart
Oh
and
man.
study the biochemical screams of anguish that the worm. This drove me into a romantic frenzy. She's like a home wrecker. It was great. So
He's
so yeah, so
wrecker.
so she and I met there. She's now an MD, PhD, MPH. I keep asking her to get a degree that makes some money. So she's a full-fledged physician scientist. She continues to work on schistosomiasis and malaria.
Wow. That
Yeah, isn't
sounds,
that a funny
that sounds
story?
pretty cool.
Yeah, yeah, it was totally fun. Totally fun.
We had
Yeah.
C.
So
elegans in space.
yes, exactly. Are you serious? Yeah, same kind of,
I
similar
did.
kind of worm.
We infected them with salmonella and E. coli. And at
Yeah.
first they were like wiggling around, probably trying to
Yeah.
get lucky with the other worms. And
Yeah.
then after we infected them, you know, we came back a few days later and they were wiggling a lot slower.
Did they mind being in space? Just being in space?
You know, they didn't, I don't know. I'm sure they noticed, but they were
Yeah.
contained in fluid, so they couldn't really
Yeah.
float.
Oh,
So
right.
they, you know, gravity was gone. They were floating, but they were in fluid. So I don't
Right.
know.
Different.
But the mice, we had mice too, and mice are absolutely, at first they're like flopping
Yeah.
around, but after a day or two, they figure it out and they start, you know, they're in these boxes and they
Yeah.
run. So then we don't have a habit trail for them like on Earth.
They
you know.
run for like Bruce
They
Dern in
floor,
that
wall,
movie.
ceiling,
Yeah.
wall, floor,
Yeah.
wall. They just made themselves a treadmill and just go like
Do,
this.
do they go fast enough to generate enough G's to actually feel like this?
Absolutely. And the thing is, you know, the NASA scientists are trying to study the effects of weightlessness on their
Right.
all muscle
Bones
and
and yeah.
but I don't think it's very valid because they're extra working out constantly and they generate
Right.
their own gravity by doing
Right.
this, you know.
Right.
So
Right.
there's
Right.
some effect, but it's not
Yeah.
what they want.
Yeah.
Anyway, it's an interesting
Yeah. Hey,
interesting
did
thing.
you get any of the eye troubles that that astronauts get?
No, I didn't, and I
Yeah.
think very few astronauts have that. It's been the big news, and I spent
Right.
most of my you know guinea pig experiment on my own
Time,
body
right.
was for eyes. But I, you know, in space you lose near vision and you gain far vision, just the way your
Right.
cornea and retina, your lens deforms.
Yeah.
But so I could see. Anyway, I'm lucky. I have good vision. I'm in my 50s. I don't need glasses, but I
Hate
didn't
you.
when I,
Yeah.
when I came. It's probably because I didn't read enough when I was in college.
Yeah. Right. Exactly. You didn't get tortured in medical school. Smart guy.
So what did your parents think when you end up getting, you know, hey, mom and dad, I got another 4 years of not getting paid, but at least it's at
So,
Brown University. What a
actually,
bargain.
my dad was stoked because he realized one of the advantages of an MD PhD program is they'll pick up the tuition for the MD years. You know, so today it's funny. I didn't tell you this. I'm actually the director of Brown's MD PhD program, the very program that I got my PhD in.
Right.
So yeah, it was funny. I'm signing my contract to become the chair of the department. You know, it's like 16 pages, and I turned to page 16, and I'm like, fuck. There it says. There it says, and you'll also be the director of the. Oh, hang on, I got a cup of coffee coming. 1 sec. Thank you. My lovely wife just delivered me a cup of coffee.
Excellent.
And it says, oh, and you'll also be the director of the MD PhD program. And I'm like, thanks, guys. Page 16, and you drop that nut on me.
And you'll be teaching anatomy, physiology, and you know, you'll be teaching orthopedics at the whatever.
Yeah, and shit that I don't know exactly. Yeah, so no, my dad was actually psyched. Turns out my father never told me this until I actually started doing the PhD. So my dad had he's one of these amazing stories of first generation hybrid vigor. His dad came over from Greece, dad and mom. He was born here, like 6 months after they got here, so it was kind of planned.
Right.
His father passed away when my dad was only 13. My dad grew up running the Greek deli. Like, you ever seen My Big Fat Greek Wedding? Like,
Yes.
that was my dad. It's actually a movie worth going to see again.
It's
really
a great
does
movie.
it still hangs. So that was my dad's life. He was amazing. Like, the idea that he crawled his way out of that into med school and then became a physician is remarkable. What he told me was, he said, when I told him I was going to work on tropical infectious diseases, he said, the thing that fascinated me the most in med school was parasitology, but I couldn't figure out how to make a living doing parasitology. And so he was really, he was actually really very supportive. Well, it's funny, soon after, awesome. I can't remember, it was one of the summers, he and I spent the entire summer in Kenya on safari. We rented a Jeep and we would go out for a week at a time, and then come back into Nairobi to resupply. So we spent 6 weeks together doing that. That was super fun.
That's awesome. Well, it sounds like your family story is exactly what makes America great. Immigrants want to come here. They start a deli, the doctor, their son is a professor. Yeah, it's unbelievable. This is what we need.
Don't we want more? Don't we want the world's best? Who would, if you had to pick somebody?
It's a competitive advantage, right?
Exactly.
It's a competitive advantage.
Why fish in a small pool when you can fish in the ocean? I totally agree with you.
Yeah, that's amazing. Well, very cool. So that's an amazing story. So, but a lot of people are interested in viruses these days, but I really want to kind of move to malaria.
Yeah.
But before we get there, just help us. And you've already used a bunch of words that I don't know because I'm just a fighter pilot. So pretend like you're talking to fighter pilots who are not Brown University quality students. So viruses in general, and we're talking about like coronavirus, right?
Yeah.
And malaria is also
fucking sick of talking about corona. Aren't you sick of it?
Yes, I'm completely tired of it.
Do it.
And you made a really interesting point about how malaria is like keeping the thumb. You said Kenya is keeping a thumb on the entire continent.
Sub-Saharan Africa,
beyond Africa. It's not just Africa, right? So is malaria a virus?
No. Yeah. So let's step back for a second. Let me put malaria into the context of coronavirus, and I do this. I'm going to be careful when I do this. I often use the example of HIV, and I equally careful when I do this. My purpose in making these comparisons is not to diminish the impact of the comparator. The comparator sucks. It's awful. It's to put malaria into the context of something that's awful, but you're familiar with. So let's do it with HIV. Yeah, let's do it with HIV for a second. So since the beginning of the HIV pandemic, which you can argue is 1984, or you could argue it's you can go back a couple of years, but there were tiny numbers of cases. Let's just say 1984. Since the beginning of the HIV pandemic, on the order of about 30 million people have been infected. That's awful, right?
That's a lot of people. Yeah.
In the first decade and a half, all of them died.
Right.
It was a uniformly fatal disease. I was a medical student during the HIV pandemic when we had literally nothing to offer, and it was unbelievably depressing, particularly for me, although, again, I don't want to diminish the impact, but for me, what was really awful was the pediatric HIV. It was just broke my heart. So horrible disease. So 30 million infections, couple of million deaths. I can't, I'd have to look up the number. Starting in 1984. Malaria has been killing between 0.5 and 2 million children every single year for the last 10,000 years. There are 200 to 300 million infections a year. Almost 2/3 of the planet is susceptible to malaria infection. They're living in malarious areas. And it kills somewhere between a child every 15 seconds and every 60 seconds, depending on a variety of factors. There truly is, if
That's
There's no
orders of
comparison.
magnitude. Right. There's a couple
Orders.
zeros. Yeah.
You got it. That's the point I'm trying to make.
Right.
HIV sucks. It's awful. Don't get me wrong.
Yeah.
Horrible.
Right.
But this thing that we don't talk about in America is so much worse.
Right.
Now let's put it into the coronavirus pandemic. And I got yelled at for making this comparison. People thought I was being insensitive. Can you imagine me insensitive?
No.
No, I don't believe it. I'm a pathologist, damn it.
We like insensitive on this show. Let's make people think and yeah.
Push them a little. No, and I'm joking. And
yeah.
Coronavirus is awful. It's decimated our elder population, and it's ruined or wreaked havoc on the lives of our younger individuals. It's awful. Yeah. However, what is the global death toll right now? Does anyone know?
It's a couple million.
Know this, I don't. Yeah, a couple million. So that's what malaria did to children last year. How many children have died of coronavirus? You can almost count them. You know, it's less than 1,000. I don't know the number, but it's a tiny, tiny number. Remember, all the crap we're doing is to preserve our hospitals and our elder population. That's why we're doing what we're doing. This is not a disease that dominantly kills children. Malaria is. Coronavirus is going to be gone in the next, give me a number, 18 months, 24 months. Right.
Yeah.
Malaria, guess what? It's going to still be here. Why? What's the difference? That's, you know, what I will submit to you is that individuals in developed countries are worried and suffer from coronavirus. Individuals in developed countries are not worried about and do not suffer from malaria. That's the difference.
If malaria had ended Europe's economy and America's economy and China's economy,
we'd be there.
You
give me $1 billion, Terry, and a year, and we have a vaccine.
Well, you have a vaccine.
Yeah, it's a good question. Yeah.
So what do we need to know about malaria?
Yeah, it's been rotting on a shelf for a while. All right, so malaria, let me tell you.
We're gonna get it. We're gonna get it out.
Yeah. Viruses and parasites.
Yes. Okay.
So viruses are really simple entities. I don't want to say life form because it's one of those silly debates in biology. Are they alive? Are they not alive? Turns out a virus all by itself cannot replicate. It needs to co-opt the cellular machinery of a host cell. And that's the distinction. Yeah, you know what? If you're dying from a virus, it doesn't make a goddamn bit of difference whether it's alive or not if it's killing you, right?
Right.
So a virus is basically nucleic acid wrapped up in some kind of capsule, sometimes with lipids, sometimes not with lipids. They're tiny little particles. Most importantly, for the most part, they have vanishingly little genetic information, which is why they can't live and reproduce by themselves. They need to use your machinery. So they're really simple. Parasite has got, 1 to 2 orders of magnitude more genetic information, and they are truly alive and will reproduce. So some parasites need hosts and vectors. Malaria typically needs a mosquito vector to transmit from host to host. And there are malaria parasites that infect birds, that infect reptiles, that infect mammals, great apes, and humans. So there are 5 species of malaria that infect human beings or can infect human beings. Of those 5, 1 causes 95% of all the misery, and it's called Plasmodium, which is the genus, falciparum, which is the species. So Plasmodium falciparum is the malaria that does the dominant amount of mischief on the planet. And you get it from the bite of a female Anopheline mosquito. So only a certain group of mosquitoes can transmit it, and only females. They're very empowered. And it turns out, I didn't know this until I got into malaria. The females need to suck your blood. Every mosquito bite you've ever gotten is from a female mosquito. And they I know. Isn't that a shocker? They're like, holy shit.
Yeah.
It's, I think,
So they're sucking your blood, but somehow the parasite gets in there?
Yeah. So the females have to suck your blood, specifically so that they can lay eggs. oviposition requires the nutrients of blood feeding.
Right.
But before they can suck your blood, imagine, you got a straw, right?
Right. Right.
Your blood will clot in that thing.
Okay.
So the first thing that female mosquito has to do is anticoagulate. Remember my day job? I'm a coagulation doc. She has to anticoagulate it's called a proboscis, the drinking straw.
Right.
Her needle.
Right.
So the first thing she does is disgusting. She hacks a loogie into you.
Right.
The spit contains anticoagulants, and for real, people are working on discovering what those anticoagulants are to use them therapeutically. One of our more recent anticoagulants is called leporudin. It comes from the spit of leeches. There are anticoagulants. You remember that great scene in Say Anything, the Stephen King movie where the kid looks down and he sees, oh, you've never seen it?
No.
Oh, you gotta see it. Great
Say
movie.
Anything. Okay.
Oh, wait, is it Say Anything? Stephen King.
The Stand.
I think it's Say Anything.
Yeah.
I think it's Say Anything. It's a great movie.
I'm taking notes here. I'm getting lesson from the professor. I'm taking notes.
It's not Say Anything. It's not Say Anything. It's the same era. Say Anything is that great movie with John Cusack, where he holds up the stereo.
Yeah, I thought that was like a funny movie. Yeah.
Yeah, Say Anything's funny. No, God, what is it? All right. Before our time is up, it's a Stephen King movie.
Okay.
Anyway, so yeah, she spits in you. The parasite is so smart. It had migrated to the salivary gland of the mosquito. So when she spits, in goes the parasite.
And this is Plasmodium
falciparum.
I'm taking notes. Yeah. I feel like I'm in class right now. I have to write notes.
Dude, it's awesome. It's awesome. You can throw the rotten tomatoes at me. So the parasite is in a certain form. It's called a sporozoid. It's a long thread-like thing.
Okay.
Enters your bloodstream. It's only there for 5 minutes. Within 5 minutes, it has left your bloodstream and invaded a hepatocyte, a liver cell. Goes to your liver within 5 minutes. There's a vaccine that's been in development for 30 years. It works great in mice. No mouse need live in fear.
Right.
Its impact on humans is pretty much worse than a bed net, sleeping under a bed net. It's designed to make antibodies, which are molecules or Y-shaped molecules that bind to things. It's designed to make antibodies that bind to that sporozoite and stop it from infecting the hepatocyte. But it has a huge problem. 5 minutes. It's got to kill its target in 5 minutes.
Right.
And in order to do that, you need unattainably or unsustainably high antibody levels.
Right. They need to be everywhere.
So, exactly. So it goes into the liver. And over the span for falciparum of between 5 and 21 days, each one of those sporozoites undergoes dramatic asexual reproduction. Each one that goes in turns into about 10,000 baby parasites. This is out of James Cameron's Aliens with Sigourney Weaver. All those little mucusy kind of creatures.
Yeah, oh yeah, yeah.
So after 21 days or so, somewhere between 5 and 21 days, the liver cells that now each liver cell that got infected has about 10,000 baby parasites. They explode. When the mosquito bites you, she'll squirt in somewhere between 1 and 30 sporozoites. So something around 300,000 or so parasites are coming out. They're called merozoites now. Each one of those baby parasites infects a red blood cell. There are people who, again, for 30 years have been trying to make a vaccine that generates antibodies that blocks the merozoite from infecting the red blood cell. And that would work. And it's an interesting approach. The challenge has been the merozoite is outside of the red blood cell for a grand total of about 140 microseconds. And so it's in. So that's one problem. The second problem is the merozoite's not stupid. Remember, this thing has been honed in the fiery crucible of evolution
Right.
I love that line. That's right up there with I'm a fighter pilot.
That that sounds like Stephen
Honed.
King. That's the Stephen
Yeah,
King.
totally. Honed in the fiery crucible.
Right.
It doesn't want to be stopped. And
Right.
so it puts on its surface variant antigens, things that look a little different each day.
Right.
And it puts up decoys. It puts up things it wants you to make antibodies to that won't slow it down at all. Clever.
This is like flying a fighter. We have electronic countermeasures. We have decoys. We have flare. We have Jeff.
I use the example from Hunt for Red October. Remember when they spew out the
noisemakers? Yeah, the noise.
Exactly.
Yeah, we have the same thing in F-16s.
Dude, one of these days, you remember the deal was you're going to get me up in a fighter plane if I do this, right?
Yeah, absolutely. Okay.
Would they ever let you take one out?
What's that?
Would they ever let you, if you just showed up at an Air Force base, would they ever let you just jump in the car?
They wouldn't let me. You can do anything once. But
right. Yeah.
I probably remember. That's my motto. You can do anything once. So, all right. So my brain is kind of full now. So there's these little malaria plasmodium, and they live in mosquitoes. I want to talk about how they get in the mosquitoes to begin with.
Yeah.
Mosquito bites you. She shoots in the loogie.
Yep.
This thing goes to the liver pretty quickly within 5 minutes.
Yep.
It does alien.
Mm-hmm.
The
goes full alien. I love it.
It goes alien. It's the I forgot what they call that thing. Jim Cameron would love that. And then it explodes, and now they're getting into red blood cells.
Yep.
And that's where the real problem comes. Do you know it when it's in your liver? Or
no? So my joke is most students, not at Brown, but at lesser tier colleges,
right?
Those students do more damage on a Friday night to their liver than is occurring throughout sub-Saharan Africa from malaria.
Like Harvard and Yale, the lower.
Yeah, exactly.
The lower two.
Yeah.
So I think why don't you just drink more and that'll clean out your liver, right?
Don't laugh. It turns out, gin and tonic, the famous, the British drink of the empire. I calculated you'd have to drink 4 gallons of gin and tonics to get enough quinine in you to protect you. And it turns out the gin is not necessary, but it makes the tonic palatable.
It's a fighter pilot challenge right there, I think.
Oh, absolutely.
But on my next trip to Africa, I know how to protect myself.
Exactly. So the parasite's now inside your red blood cell.
Right.
This is where the damage starts. Each merozoite, remember 300,000 came out of your liver.
Mm-hmm.
Each merozoite enters one red blood cell and undergoes asexual reproduction. It goes alien again inside your red blood cell.
Yeah.
One, over the span of about 48 hours, one baby merozoite enters, turns into this amoeboid looking thing called a trophozoite. Then it divides its nucleus till there's about 20 different nuclei in there. Then it segments into individual merozoites around those nuclei, calling that thing's called a schizont. And then it ruptures, releasing about 16 to 24 merozoites, each one of which goes on to infect another red blood cell. I joke I used to do this with the students, but then I got too old and they're too young. Do you remember the Joe Namath Fabergé Organics commercial? They tell 2 friends and they tell 2 friends. Yeah, exactly. That's geometric progression. That's what's going on.
They don't know what you're talking about. So in the F-16,
they look at me like, we had a bomb called a CVU-87. We called it the 100 miracle bomb because 100 miracles had to happen for it to work. It dropped, and then it had a radar altimeter, and it would go to the target, and then when it got to a certain altitude, it would split, and then all these little bombs would come out, and then they had special sensors. They could tell if they hit the ground or if they hit a tank, or it was like 20 things had to happen for it to do its damage. And this over-engineered.
Yeah, this parasite sounds like that. There's so many different levels, and they're so completely unrelated, and they're so damaging.
It's an incredibly difficult adversary. There's no question about it.
Yeah.
The sort of military analogy truly holds. It's a true enemy. I don't know if this is true in your prior experience, but commanders in the Army used to have these things called coins. And if you didn't have your coin on you, you'd get yelled at or whatever.
Of course, yeah. Yeah.
So there was a famous malaria scientist, physician scientist in the Army, because the Army wants to get rid of malaria big time. I can explain why in a little bit, but he had a coin minted with the Latin "Malaria delende est," malaria must be destroyed, which was a great coin. I'd love to get a copy of that coin.
I love that.
So yeah, horrible. It's a horrible adversary.
Yeah.
So remember, as these cycles are going on in your red blood cells are bursting.
Right.
Red blood cells are critical for delivering oxygen to tissues.
Yeah.
And when you don't have enough red blood cells, you're not delivering enough oxygen to tissues, and your tissues die. And one of my favorite organs needs an awful lot of oxygen.
Right.
And so
your hair?
Yeah, exactly. My hair. Yeah. Dude, 53. The only good thing God gave me, good head of hair.
That's impressive.
So this is where the disease happens. It's not just red blood cells getting ripped apart. You have an organ in your innards called the spleen.
Yeah.
And the spleen is designed to filter blood. And it's basically, the Melita filter of your blood. It would filter out and kill parasite infected red blood cells. They look a little weird. They don't deform properly. Your spleen would get rid of infected red blood cells.
But not all.
No, exactly. And if you're a parasite, you want to avoid the spleen at all costs. Mission critical. Don't go near the spleen. Turns out, here's your red blood cell. The parasite's inside. It's actually inside a sac. When the parasite enters the red blood cell, it does this invagination of the red blood cell membrane. So inside the red blood cell is an inside-out vesicle made from the red blood cell membrane. Inside that is where the parasite is. It's kind of crazy. Here's what the parasite does. I want to avoid the spleen. From inside the sac, which is inside the red blood cell, it makes specific proteins. I call them Velcro. And it puts these Velcro proteins on the surface of the red blood cell. So it gets them across the parasite membrane, across the inside-out membrane, through the cytosol of the red blood cell to the red blood cell membrane, and gets them on the outside of the red blood cell membrane.
Okay.
Now you got a parasite in a red blood cell that's got Velcro on it. And the Velcro sticks, hook and fastener, it sticks to your vascular endothelial cells. These are the cells that line your capillaries.
Okay.
And so by sticking in your capillaries, in your arms, in your legs, in your lungs, in your guts, in your brain capillaries, you avoid traveling to the spleen. But if you stick in the capillaries of your brain, we call that something. We call it a stroke.
Right.
So you get cerebral malaria. If you stick in the capillaries of your lungs, you get pulmonary malaria. If you stick in the capillaries of your gut, you get GI malaria. If you stick to your placental bed, you get pregnancy malaria. These syndromes cause severe disease because it's like a Drano problem. Once a bunch of infected parasites have stuck in the vessel, regular red blood cells can't get through. And so any tissue downstream that gets its oxygen from that blood vessel will die. So it's not just that you can't deliver enough oxygen because your red cell number is dropping. It's that you can't get the oxygen you do have delivered on site because you can't get through the pipes. So it's a really bad disease. And this is why it's so
unbelievable,
fatal.
Jake.
Yeah,
it's
crazy.
It's not like a virus that clogs up your lungs or clogs up whatever, your heart. And it's like a complicated weapon system.
It's totally. It actually makes you either believe in a malevolent deity,
right,
or really buy into the theory of natural selection. This thing is, honed in the fiery crucible of evolution.
Right. That is unbelievable. Wow. So how does it go from mosquito to mosquito?
Yeah. So every once in a while, everything I've discussed has been described as been asexual reproduction, and the parasite gets bored and wants to get it on once in a while. And so it actually differentiates in the blood. Every once in a while, it'll differentiate into a male and a female gamete form called a gametocyte.
Right.
So male and female gametocyte. They look like bananas. They're banana-shaped forms. Another mosquito comes along. Oh, and these banana-shaped forms, They hide in your bone marrow until they're mature, and then they come out and they tend to stick in your skin capillaries. It's freaking
Where the
design.
mosquitoes go.
Ding, ding, ding,
This
ding.
is design.
Yeah, it's totally. This is designed by evolution, though. Design. Remember, the parasite that did that, the first parasite that did that, had a huge selective advantage. It would get picked up by the mosquito.
Yeah. Okay. Yeah, but it goes to the capillaries to get sucked out by the next mosquito, right?
Right.
So
Isn't
it
that
uses
awesome?
the person to grow the little alien pods, and then
We're petri dishes, man. So mosquito comes along, takes a first blood meal. The male and female gametocyte get taken up in the blood meal. In the belly of the beast, they find it so romantic. It's, Liberace with the candelabra and the piano music. They conjoin, turn into an ookinete. Now it's a fertilized egg, basically. This thing burrows into the gut of the mosquito. Over the span of several weeks, depending on the temperature, because remember, a mosquito is an ectotherm. It doesn't regulate its body temperature.
right.
So depending on temperature, and this is where climate change comes in. As higher elevations get warmer,
right.
You got it.
Yeah.
So burrows into the wall, differentiates into baby sporozoites, crawl up through the hemothorax of the mosquito to the salivary gland. When the mosquito takes her next blood meal, in they go.
This thing is alien. This thing is alien. It kills the host and uses the host to grow itself for the next generation.
So, as it turns out, and we're going to come back to what you just said, it kills the host. Believe it or not, it is not in the parasite's best interest to kill you. It wants, when I speak teleologically, it wants to grow to the maximum extent possible in you without killing you.
And it doesn't kill adults, right?
Tends not to. Adults become resistant. If you or I went over to Kenya right now and walked around unprotected, we'd die like dogs. You want to hear a funny story? True story.
I'm sure it's really funny.
So there I am. This is 1996. I'm in Kenya with my postdoctoral advisor, and we're enrolling a cohort of Kenyan children.
Right.
And I still work with this guy, which is surprising after I tell you this story. So I'm starting to get a little sick, right? And getting a little woozy, and we're doing physical examinations on these children.
Right.
And I tell Patrick, my postdoc advisor, I say, Pat, I don't feel so good, Patrick. And he looks at me. He's a doctor too. He's looking at me, and I'm all sweaty and pale, and so he decides to take orthostatics, which is, if you're worried someone's dehydrated, you take their blood pressure laying down. You sit them up, you take their blood pressure. You stand them up, you take their blood pressure. So he stands me up, and I go down. Boom. And I'll never forget. He didn't catch me. It was this lovely Kenyan nurse. She was a big, strong woman. Grabs me on the way down. So it's a long story. The physical portion of the exam I was measuring Tanner stage. I was assessing pubertal development of these kids, which basically was, it's kind of a subjective exam measuring how well developed they are. And you don't want a lot of scientists, observers doing it. You want one observer doing it. But even though I was sick, he made me lie down on a cot and finish the exams I was doing. I was like, thanks, pal. True story. So the idea is, do I have malaria or not? So we go the next day to the world's best microscopist, the Kenyan microscopist named Ramadan Matalaba. And we go to Ramadan, and he spends 45 minutes looking at blood films from me.
Mm-hmm.
And I'm laying on the cot, rigoring.
Right.
And finally he says, Bona daktari, kuja hapa, come here. And so I look at the microscope, and there on this microscope is a single, now he's been looking for 45 minutes, a single mutilated looking parasite. And he looks up at me and he goes, Bona daktari, you white people, you're so weak. It was awesome. Because if I were a Kenyan, I would have thousands of parasites and I'd still be working.
Right.
So my point being, there was a logic to this arc here. The point being, if you don't have acquired immunity, like you or I, if we went to Kenya, would have no acquired immunity, we would drop like dogs from the first parasites we see. If you live in a holoendemic area and you survive, over time you will develop protective immunity.
Right.
So
I've been thinking that all this hand sanitizer and wearing masks and stuff, it seems like that's going to make us weaker for other potential problems.
No doubt. Yeah. Under the circumstances, though, you got to do it. But
no, I understand. Yeah, but it is.
I mean, have you seen? There've been essentially no cases of flu in the United States this winter.
It's great.
It's
I get a cold every year or two, and I've been completely healthy.
Nothing. I know.
It's good. We did that same orthostatic test after I got back from space.
How'd you
do? The blood pressure is a big deal because you're dehydrated, and they made us do a push-up and then stand up as fast as you could. And it was awful. It was terrible, but I was able to do it. It sucked.
No one needed to catch you.
No, I always wanted someone there because it wasn't like orthostatic intolerances for the fighter pilots out there is when you when you get up too quick and then you're like, whoa, yeah.
But you guys learn how to Valsalva when you're pulling G's.
Oh yeah. Well, no. Not Valsalva is when you change pressure.
Yeah. Oh, what do you guys do when you're pulling a G turn? You guys do something.
It's called an anti-G straining maneuver. So you just tighten the muscles in your legs and especially your core, your stomach.
Yeah.
And that, you don't want to do it. Don't try this at home, but I can turn myself red by pushing blood up into my face.
Right.
And because when you're pulling G's, the blood gets sucked out of your brain.
Right.
Anyway, sometimes if I stand up and I get dizzy, I just do that real quick and it fixes it.
Yeah. Your natural tendency. So bad parasite. People have come up, they're trying to do transmission blocking. Imagine I vaccinated you to make an antibody that's directed against the parasite form that only shows up in the mosquito. She takes her blood meal, the antibody and the parasite come together, right?
Mm-hmm.
The antibody has nothing to bind to until the gametocytes come together and form the ookinase. Now the antibody that's in the blood meal attacks. It wouldn't protect you from your malaria, but it would prevent you from transmitting it to somebody else.
Is that your vaccine?
Nope.
Is that the other one?
That's that's actually a guy that my former MD PhD student, who's now my faculty member. I shouldn't say mine. He's his faculty member.
Right.
That's the kind of vaccine he works on.
So let's talk about the vaccines. So and the history of failed vaccines and why yours is the best.
I don't know about best, but
I know we're running out of time, but yeah.
I'll say I'm agnostic. I really like mine, but and mine is really ours. There's a huge group of people that did this work.
Right.
But I think fundamentally, people thought malaria was going to be easy. Once we got a few molecular tools on board in the, in the late 80s, mid 80s, we thought it was going to be easy. We knew that we could protect mice if we gave them irradiated parasites, mouse parasites to mice, but the parasites were irradiated. They'd get to the liver and then die. Those mice were protected from a new batch of parasites. Those mice made antibodies to a particular target. The target was the sporozoite. Works perfectly in mice. For 45 years, we have had an outstanding vaccine for mice. No mouse need live in fear. But the generalizability to humans has been atrocious. It doesn't seem to work very well in humans. It works less well than a bed net.
Wow.
So my point being, if you start with mice, you will certainly protect mice.
Right.
It's a different host and it's a different parasite. Human parasites do not infect mice. So the reason people start with mice, I'll be really honest, it's easy.
Yeah.
I can walk out, I'm in my laboratory office. I can walk out there are my mice. Yeah. There are my parasites. Easy, easy, easy. You can answer many questions quickly.
Right.
I submit to you, much harder is to put on your PIF helmet and waders and go into the field.
Yeah.
It's dirty in the field. And I mean that scientifically dirty. It's complicated. It's messy.
Right.
You can't control everything. It requires extreme epidemiologic planning. You need different kinds of scientists. You need epidemiologists. You need biostatistics. Then comes the molecular biology and immunology. So it takes a village of scientists to study a village of people with malaria. But what I can tell you, Terry, is if you go into any village in a holoendemic area, you will see a natural progression in those human beings. Very young children are very susceptible. Middle-aged children still get parasites, but they don't get sick and die after the age of 6 or 7. And by the time they're a little bit older, they're not getting as many parasites anymore. So resistance comes in 2 phases: anti-disease resistance first, then anti-parasite resistance.
Okay.
Why not figure out what the hell is going on in humans?
Right.
Instead of starting with mice. It sounds obvious.
Right.
And yet, no one had reduced to practice or reified this approach where you combine epidemiology and cutting-edge molecular biology to identify the parasite genes that encode parasite proteins that are uniquely recognized by resistant children, but not by susceptible children. The secret sauce, Terry, is that susceptible children make antibodies to parasites. Almost the same antibody repertoire. If this is like Venn diagram, if this is the antibody repertoire that susceptible children see, and this is the antibody repertoire that resistant children see, they're not that different. They're like this. Resistant children see only a tiny fraction of additional targets.
And that's the key.
That is the secret sauce. Those additional targets, the Boolean difference product, that's what we did. We determined the Boolean difference between susceptible and resistant kids in terms of what parasites they target. So, you're bringing this back to an adversarial sort of paradigm. If I send in antibodies against your hair, you're going to just shave your head. You're going to go Telly Savalas. If I send in antibodies to your heart valves, you're screwed.
Right.
So the difference is the heart valve targets. What are the important targets? What are the targets that children over time, if they survive, develop protective antibody responses to? And that's what we did.
And that requires a lot of discipline over years to take the data and go into the field. And
5 years of field work following, and this is where Patrick Duffy and his scientific partner, Mikhail Fried, and their team, always remember the team.
It's always a team, right?
It's always a team. It's always a team.
Like space travel.
Exactly. Yeah. You're not getting there alone. They did a birth cohort. They followed a birth cohort of almost 1,000 women giving birth to their children. And they followed those children every 2 weeks with blood films over 4 years. And, roughly 47,000 blood films, which is how you diagnose malaria. And using that information, we could figure out who got more malaria, who got less malaria. Now, to a first order approximation, you would say, if you're getting more malaria, you're susceptible. If you're getting less malaria, you're resistant. Except, what if the guys getting less malaria slept under bed nets?
Have less mosquitoes, right?
You got it. What if they had Sickle trait, which is a hemoglobin variant that protects you from malaria? There are a whole slew. What if they took antimalarials? So the epidemiology piece is remove those non-immunologic factors. What's left over, the protection you see that's left over, a portion of that is due to antibodies. And that's what we did. We figured out what are the parasite genes that encode parasite proteins that only resistant people see. And that's turned into a short list of vaccine candidates, some of which we've really pursued. And my favorite one is this protein called GARP, glutamic acid-rich protein. It was also my favorite novel as a kid in high school. I did write to John Irving, and he still has not written back. I'm pissed.
Bastard.
And you know what's funny, man? Have you ever written to an author before? Like, It is.
I have written to Tom Wolfe, and
No way.
I got in touch with his literary agent because I flew the DVD of The Right Stuff on my first
Yep.
space shuttle flight, and I was going to try and set up one of my trips to New York City, and unfortunately he passed away. So.
Oh, I'm sorry to hear that.
Yeah.
No, I didn't. So when I wrote to him, like, it was the most carefully written letter I've ever constructed. You know, it's like you're writing to a literary genius. You know.
Right.
Anyway, I'm still waiting, John. Okay. So.
Well, hey, real quick, Jake. So sometimes
yeah.
people will write to me. Anyway, he may just not. You may make sure you're sending it to the right address. But anyway, go ahead. We'll continue.
So GARP.
Yeah.
GARP is a parasite protein that's put on the surface. Remember I told you about the Velcro that's up on the surface of the red blood cell?
Right.
GARP's on the surface of the red blood cell, but I don't think it's involved in sticky. What I can tell you is when antibodies bind to GARP, the parasite inside the red blood cell commits suicide. It undergoes apoptosis, which is programmed cell death. Why would a parasite put a kill switch up on its surface?
I don't know.
Here's the thought. I don't know the answer to that question.
Right.
And to a certain degree, I actually don't care what the answer is.
Right.
As long as I generate antibodies to GARP in you, you're going to be protected.
Right. And some kids do that, right? Like some kids have that.
The resistant kids do.
The
resistant kids do.
Yeah. Right.
You got it.
Right.
GARP was one of the differentially recognized parasite proteins.
Right.
So here's what I think is happening. The parasite wants to multiply to the maximum extent possible.
Right.
But not further.
Right.
So how does it know?
Yeah.
That was an Einstein quote, you know?
Yeah.
Things should be explained in the simplest way possible, but no simpler.
No simpler. Exactly.
Right? So how does the parasite know? One possibility would be to sense how many other parasites are around. The problem with that is some people can tolerate higher parasite burdens than others.
Right.
So if you set a threshold too low, that would not be your evolutionarily maximized growth density. A better way would be to sense when the host is tapping out on the mat.
Mm-hmm.
You got the chokehold on. When do you tap out?
Right.
When are you sick? So you're sick when you have a high fever, and that high fever is mediated by molecules in your blood that one could imagine measuring. So my pet hypothesis, and it's truly just a hypothesis at this point, is GARP is sensing the health status of the host. When the host is in deep caca, it activates parasite death to shut down parasite replication. So we would call that a density-dependent regulatory mechanism. And you're going to love this. Ready for the full circle rainbow?
I'm ready.
The sea urchin project I did when I was 21 years old or 20 years old.
Right.
The title of the paper, Density-Dependent Regulation of a Rock-Boring Sea Urchin.
Wow.
The same fundamental concept is what I think is happening with GARP. Why does the parasite have GARP? It would be such a sweet, circle of life.
I want to do a documentary about this.
Oh, no way. That'd be fun.
Yeah. I mean, this has been my thing I want to be a filmmaker and I want to make a documentary. So we're going to have to get this on film. So this, okay. So you spend your years in Africa, you're studying these kids. You're like, wow, some of these kids are resistant. You said something really interesting. You said the sickle trait. Sickle cell anemia is a problem that I think mostly Black people have that is deadly, but ironically, it can also protect them from malaria.
Remember that whole fiery crucible of evolution thing?
Yeah.
Guess what? It applies to humans. So human beings bear the genetic imprint of coevolution with parasites. In particular, they bear the imprint of coevolution with falciparum malaria. Sickle trait is the common one. So hemoglobin, you have is a complex molecule. You have alpha chains and beta chains that come together around a central inorganic molecule called tetrapyrrole, or the heme group, and it allows the binding and transport of oxygen.
Right.
Alpha chains and beta chains. If you have a single amino acid mutation in position 6 of the beta chain, and remember, you get one copy of the beta chain from mom and you get one copy of the beta chain gene from dad.
Okay.
If one of those two copies has that mutation, you have what's called sickle trait hemoglobin.
Right.
Zero disease as long as you're not at altitude. If you're unpressurized, you can actually have a sickle crisis. But
right.
If you're normal pressure, I love how I'm bringing this back to fighter pilots.
Yeah,
So
I'm
wouldn't.
listening
It
now.
would not surprise me if you guys get tested for sickle trait. It would not surprise me.
We might. I don't know.
So if you're sickle trait, you have a significant protection from death due to malaria.
Wow. But you might die from sickle cell anemia.
If you get two copies, you're hosed.
Okay.
It is in the 1990s, it was uniformly fatal in sub-Saharan Africa, uniformly fatal before procreation, before the age of 12. Now, in some parts of sub-Saharan Africa, there's therapy. In the United States, up until 5-6 years ago, you would be getting, highly variable disease. Some people have very bad disease, some people more mild. Typically, lifespans in the 30 to 50, sometimes beyond year range. One of the slickest things, you want to get someone else on this podcast to do cool science, gene therapy for sickle disease. If you look up, there's a lot of film on this woman. I'm sorry, I can't remember her name. You'll find it if you Google.
Okay.
She has been successfully genetically modified.
Wow.
Her sickle disease, which was incapacitating,
Right.
is for her cured. So you're going to see that come out, out of the research clinic into the standard clinic in the next 5 years for sure.
We'll have to get her on.
Here's the thing to really remember. Uniformly fatal, that means you're not procreating. Your genome is going into the toilet.
Right.
How much benefit must there be from the hybrid state, sickle trait, in order to tolerate the complete obliteration of the homozygous deficient trait, the homozygous deficient state? The people with sickle trait, one copy, have to have such a selective advantage that the gene stays in the population. That selective advantage is because they don't die from malaria.
That's unbelievable.
It's
so
crazy.
I think what you just said is if you have partial sickle trait, you're protected against malaria, which helps you out. And there's more kids because they survive better. But if unfortunately you get both mom and dad give you it, then you die. You
die.
Amazing. But these are people, these are human beings. This is like interesting biology for the Brown University professor, but these are people.
It's awful. It's awful. It's awful. I will tell you, working in the field is the greatest motivator because you connect the actual human suffering, which is incalculable. Let me put it to you this way, Terry. So imagine a society that, some of the places where we work, the under 5 mortality rate is 25%. Imagine a society that has suffered the indignities of colonialism, followed by the indignities of dictatorships, followed by the indignities of massive under 5 mortality. You show me a society like that is able to capitalize on its potential. It's not going to happen. It's not. And so I really do believe, those other two issues are huge issues that happens not to be my professional expertise. The third issue is something that I think I might, I've devoted my life to trying to do something about.
I know we're getting short on time. Talk about, so the GARP protein is kind of your secret sauce. You found this protein, it works. So how are you going to get that in shots into arms? I think money is the answer, but
I hate to say it, man, it's greenbacks. So we have known about the importance of GARP. We cloned it in 2008. We really figured out, we were working on another protein between 2008, 2012. We were working on SEA, which is our second. It turns out it's the one we moved first with, but we have 2 vaccine candidates that are both pretty slick, and we think they'd work great together, as it turns out. So really in 2012, 2014 is when we really did the key work on GARP. Since 2016, I've known this is the one to move forward with. It took 4 years from the time I knew it was the one to move forward with until I was able to get it published in, the fancy magazine.
Right.
So this came out 6 months ago.
That's big time. Yeah.
But all right, it's a bunch of PhD academic bullshit until it turns into shots in arms. So quick funny story, and it connects to COVID. In 2017, I'm giving grand rounds down at UPenn, and I'm giving the talk, and there's a guy in the audience asking really tough questions, and which
that
is
guy.
all good. It's that guy.
It's always that guy.
He was super polite,
right.
No issues. He was really interested in the topic.
Right.
And after you give grand rounds, normally you meet with people who signed up to sort of have a meeting time with you. So I whipped through this lab and walk into an office, and there's the guy. His name was Drew Weissman. And Drew started telling me about this new vaccine platform he had developed. And the way he was speaking, I thought to myself, the guy is either brilliant or he's the biggest BS artist I've ever heard. It turns out I read his papers. He's brilliant.
Right.
What he discovered is amazing. A brand new platform. He was telling me about a little difficulty he had getting funded because he was so avant-garde.
Right.
He was having some trouble getting funded. One of his research associates, a Turkish woman, had to leave his group. She went back to Germany to start a small biotech company. The name of the biotech company you might be familiar with, it's called BioNTech. The platform was lipid encapsulated mRNA for vaccine delivery.
Right.
The Pfizer BioNTech vaccine is built on Drew Weissman and his team. Platform. So when I got my Pfizer BioNTech COVID vaccine, I sent him a text saying, Drew, thanks a bunch, pal. I feel the antibody power coursing through my veins. He convinced me, Drew convinced me during that meeting to spend the last $250,000 I had for a monkey study using his platform, which when I look back was a crazy gamble. If you look in our Nature paper, Drew Weissman, one of the fathers of the Pfizer BioNTech COVID vaccine, is an author on our malaria paper. Our vaccine platform is predicated on the lipid mRNA technology. The risk back then in 2017 was the FDA didn't know how to regulate this platform. Guess what? With $1 billion in a pandemic, they figured it out.
Right.
So we have a flight plan directly in front of us. We know exactly what we need to do. We need to do 3 or 4 more monkey studies, and we need to do our phase 1 studies. The only thing keeping us from doing that is financing. NIH likes to discover vaccines. They don't like to develop them. It's not in there. It's not what they do. So
you're ready for
what's called the Valley of Death.
You're ready for phase 1 and phase 2 trial. You do it on a few people, then you do it on some more people, and you need money. And that money is probably in the millions. It's not hundreds of millions, but it's millions of dollars.
Tens of millions gets us to a phase 1.
You need tens of millions. Okay. So, and we've talked about this. We have strategies and stuff. We won't go into detail, but if there's any finance type folks listening to the podcast, get in touch with me, get in touch with Jake.
Dude,
we will
open up the wallet, man. Send it out.
Well, it's going to change the course of human history, and it's going to change the course of a continent.
Whoever develops the malaria vaccine will have made a fundamental, and it will be a team of people, whoever that is. And I actually don't care who it is, as long as someone gets around to doing it, will change the course of human history.
Yeah, in a profound way. Coronavirus is massively important. It's killed way too many people, and it's a couple year problem. Malaria is a 10,000 year problem.
Exactly.
And it's never going to end. This is never going to end.
It will not end until we get better tools. We need
intervention.
Better tools.
Right. Okay.
And I know if this isn't the moment with coronavirus of science, triumphing over darkness, I don't know what is. We need to extend that momentum. We need to have the political will to actually care about our fellow human. And, I pitch this even to the business types. The business types, they need markets for their widgets.
Mm-hmm.
Whereas you're never going to get the sub-Saharan African market until we help raise them up out of the economic pallor. And so you can even make a selfish argument that in order to generate those emerging markets, you need to actually get health in line first.
Right. You need to make a selfish argument because then people will be motivated to finance it. and I kind of don't care why. I just
I don't
care
either.
that,
Yeah.
right? That
I
it
care
happened.
when, like
Yeah,
now.
right. Right. Okay. Jake, this is unbelievable. This is so cool. Honestly, I never thought in a podcast I would talk about Boolean algebra.
Isn't that funny, man?
This is pretty amazing. I wanted to talk a little bit more about COVID and was it a lab leak or not, but maybe we can do that the next time.
Yeah. Next cycle.
Yeah. All right. Well, thank you so much for your time. This was fascinating, and this is a problem that we need to fix. So we need to figure out how to get this financing done. I think you might be getting it done. I hope, but let's
fingers crossed.
Let's make sure this happens. And millions of people depend on it. Hundreds of millions of people depend on this.
Crazy.
And so it's really important, and it's never going to go away until we fix it. So, all right. Well, thank you for this. If you guys have enjoyed the podcast, please give us a rating and subscribe to the podcast, and hopefully we'll be in touch, and I'll make it out to Brown one of these days soon.
We would love to host you for sure. For sure.
That'd be fun.
That's a good thing to look forward to after the summer misery is over.
Yes. All right. Well, thanks,
Almost
Jake.
there. Peace, guys.
Talk soon.
Take care.
All right. Bye-bye.